Good afternoon. My name is Jose Daly. I will be doing a review of effects of climate change on orchid population. Introduction. Over the past 200 years, Earth's climate has been transformed by human activities by rapid development and industrial evolution. However, orchids' current climate change is quickly increasing in temperature at an unnatural rate. Habitat degradation is increasing, shifts in weather patterns are generating substantial precipitation, increased drought, rising sea level, and a rise in invasive species are damaging the security of orchid populations. Climate change reduces a population's durability and creates a crisis in diversity, pollinator accessibility, and expansion. The objective is to comprehend how orchid populations are distributed and enduring climate change's influence. Climate change. In the next coming decades, global temperatures will increase between 1 to 5 degrees Celsius and considerably alter environmental factors throughout the planet. Stochastic events such as droughts, wildfire, and hurricanes affect populations and are only becoming more frequent with climate change. Increasing temperatures and radical weather events are diminishing the ecosystem's ability to regulate water flows, improve water quality, simulate natural cycles and interactions, prevent extinctions of beneficial species, and accelerate expansions of harmful species. Orchid species. Over 70% of orchid species are epiphytic, found on tree trunks and branches. Most epiphytes are found in South and Central America, Mexico, the Pacific Islands, and Asia, while terrestrial orchids are found mainly in Europe, New Zealand, North America, and Australia. Variations in local climates begin to force native species to respond to avoid extinction. Certain species must adapt and change their most change their physical characteristics as well as behavior in addition to preserve harsh conditions. If these species are able to adapt, then relocation is the best alternative compared to perishing. Many geniuses and species are found throughout the globe and are known to withstand intermediate climates. Figure 1 shows the silology for many different variations of genetics that result in different species and outgroups. Orchid populations. Depending on the location and silology of the population, they are bound to respond accordingly by migration, adaptation, or competition against other populations or face local extinction. The distribution and abundance of orchid populations depend on a suite of biological, and ecological factors, including seed production and dispersal, availability of mycorrhizal fungi, and appropriate environmental conditions, with the weighting of these factors depending on the spatial scale considered. Abundance of populations mainly depend on their ability to comfortably influence environmental factors that establish plant growth, seed dispersal, and interdependence with pollinators and nitrogen fixing bacteria. Abiotic factors. Abiotic factors such as climate, soil quality, and luminosity influence how orchids react to their environment. Soil biochemistry and climate are the most vital factors for distribution of orchid species. The ecological niche of orchids is the principal attribute to an orchid response to soil composition, temperature, precipitation, altitude, vegetation category, and bedrock. Other factors such as habitation fragmentation, pollinator decline, human activities such as poaching are greatly affecting orchid populations. Biotic factors. Biotic factors such as symbiotic relationships, competitive species, and vegetative growth impact the development along with forthcoming growth. Pollinators are extremely crucial for all types of vegetation. Orchids are advantageous with many different species, such as wasps, bees, ants, flies, moths, small birds, and mycorrhizal fungi, assisting in cross-pollination. With an increase in temperature, pollinator species are suffering. As temperatures increase, sugar content in nectar is steadily decreasing, which decreases its nutritional value. Migration and patterns and being altered, and pollinator populations are coerced into following these disruptive changes. The timing of seasons are slowly shifting to later periods of the time, which divert pollinators from accurately pollinating for later migration or hibernation. These changes are making ecosystems unstable, which in turn disrupt pollinator interactions. Propagation. Propagation is the natural procedure of increasing the number of plants. There are two main forms of propagation, sexual and asexual. The most common form is sexual propagation and involves the creation of seed capsules and the release of pollen, while asexual propagation involves a division of pseudobulbs and rhizomes, which is when overgrown orchids are divided into separate plants. Micropropagation is the act of using minuscule plants of plant tissue from a mother plant and grown under a laboratory environment, producing a new proliferate and disease-free plant. This technique is mainly used to minimize the pressure for threatened and endangered wild populations. Intro vitro propagation is a specific method of micropropagation in which orchid mediums are added to endangered species in order to conserve the species. Mediums such as wood bark, coconut water, potato dextrose, horticulture charcoal, banana powder, and even sphagnum moss are functional and affordable for the alteration of jeopardized floral species. Figure 2 shows the future predictions for temperature change for the upcoming 80 years based on different scenarios of human emission pathways. The red line assumes humans will continue to emit carbon dioxide. The green line assumes humans will balance non-renewable and renewable resources. The blue line assumes humans will make an effort to reduce non-renewable emissions over time and switch to sustainable choices. Figure 3 shows the changes in microbial community during plant range expansions, with graph A showing the span of range expansions for plants, while graph B shows the hypothetical expansion of plants affected by pathogen and bacteria communities. Figure 7 shows the degree of dependence for each orchid species occurrence in the corresponding habitat. A study composed in the Department of Biology, Division of Plant Conservation and Population Biology by Catholic University Leuven in Belgium focused on the current and future impact of climate change on the distribution of 4 European terrestrial orchid species. The high degree of niche similarity was reflected by the variable contributions output, with the 4 species reflecting similarly weak associations with bedrock, elevation, and 2 of the soil components, with being more affected by the first 2 soil components, pH and potassium level, and soil texture, temperature, and precipitation. Biotic factors. Figure 7 shows an example of pollinators, such as the orchid bee interacting with endangered orchids. Figure 7 shows the germination percentage of a selected orchid species, the night-scented orchid, after a period of time while sowing the seeds supplemented with the mediums. Graph A showed a time period of 30 days. Graph B showed a time period of 60 days, and Graph C showed a time period of 90 days. The variables indented in the graph are CW for coconut water, BD for potato dextrose, BP for banana powder, C for unaltered seed as a control, and BP plus CW for a combination of banana powder and coconut water. For each trial, coconut water exhibited the best outcome for seed germination. Prospective solutions. Figure 7 shows the red list from the International Union for Conservation of Nature. Graph A reveals the future research priorities, while Graph B reveals the conservation priorities for endangered and threatened orchids. Prospective solutions. Conservation techniques such as monitoring and managing orchids, seed storage and banks, habitat and ecology maintenance, and the assisted migration of wild orchids and relocation of propagated orchids to restored habitats are among the most important modern-day orchid conservation techniques. Orchid regeneration is the goal, with large-scale ecosystem restoration in mind. This involves soil and vegetation monitoring, reintroduction of common plants and animals, procure volunteers or employees to care for the habitat and the organisms within, identification of the deprived areas in the need of conservation, evaluation and overseeing the amount of human interaction, and undertake sustainable applications. Many research organizations and programs such as the North American Orchid Conservation Center, NAOCC, RBGV Orchid Conservation Program, Orchid Conservation Alliance, American Orchid Society, AOS, Care for Biological Diversity, and South Florida Orchid Society are working diligently to find new species, successful viable solutions, and safe environments for orchids. Here are my references, and thank you.